This is Lab 10, Selective Media number 3. We're going to be using McConkey agar plates to investigate what kind of bacteria are present in water samples. So you're going to need to get 1 or 2 water samples from a local park to do this lab. Like usual before lab, we want to disinfect surfaces, wash our hands, and wear the appropriate PPE. So this is the Selective Media experiment number 3, and I have already poured my McConkey agar plates. You're going to need 3 of them in total. 1 will be a control, and so the control is just your distilled water like we've been using. I have to go grab mine because I forgot to do that. And then you're going to need 2 water samples that were described in the procedure to go collect. So some body of water in your community that's not treated with chlorine. So something like at a park that has birds and things that are playing in there. If you want to get 2 of those, you can, or you can just get 1 water sample and then the other you can test as a kitchen sponge or something like that, or a dish rag. So I actually have 2 water samples, one from the canal that's close to my house and then one from a park. So those are the 2 that I have. So those are going to be the 3 plates that you're ultimately going to plate. And like before, we're using our pipette pump along with our pipettes. So it's just going to be some drops on the plate, and then you're just going to swirl it around to basically spread it. So you're not going to use the spreaders this time. Okay, so now we're going to plate this out. Earlier I said distilled water, and I was looking at the procedures. The control should just be tap water. So this tube has just water from my kitchen tap, and so those are the 3 that we're going to plate. So just make sure you have the right tube corresponding to the correct plate. So this is the control that I'm going to do first. So it just asks you to do about 4 drops onto so that's the control, and then just give this a little swirl to make sure it's kind of completely coated. Okay, so that one's done. The next is the center one. This is the water that I got from one of the canals near my house. So same procedure, 4 drops. So again, just make sure you're putting it on the right plate. And then after you pour or put those 4 drops on there, you just want to spread that liquid around. So that's good. And then we have 1 more to do. And this is from a pond at a park close to my house. So again, 4 drops. And make sure that's spread. And as you won't be able to see it on camera, but I can kind of see where that film of liquid is on the surface. So I'm just trying to make sure there are no kind of dry spots and that the water that I just put onto the plate is completely distributed across the surface. And so once that has occurred, that's good. And as we've done with all of our experiments, we're going to parafilm each of these plates and then put them to incubate for a period of time. And then you're going to be looking at these over the next several days to look for growth. And so the next part of the video, I'll show you what they look like after they grow. So after the lab's done, you're going to parafilm your plates. Make sure to take a photo and then disinfect all the surfaces you've been working on, and then also wash your hands. We are done with the water samples in the tube, so you can discard those.
And these plates are going to incubate somewhere between 3 and 5 days prior to you evaluating them. So just keep an eye on them, and then the next part of the video will explain what to look for. So this is a short video to explain how to evaluate McConkey auger plates. And what's important to know with McConkey auger is that it is two types of media in one. So it is both selective media as well as being differential. And so remember what selective media means. It means that any selective media is going to cause or allow certain bacteria to grow while others are going to be inhibited. And so McConkey auger, the way that it works is it has both bile salts and a dye called crystal violet. And the presence of those two items is going to inhibit gram-positive bacteria from growing. And so what that means then is if something is growing on a McConkey auger plate, it's going to be a gram-negative bacteria. And so we can say this media selects 4 gram-negatives or gram-negative bacteria, and it's going to inhibit gram-positive bacteria. So that's one thing that you have to keep in mind. And then second, is it's also a differential media. And what that means is there's going to be something in the media that allows for a color change reaction that's going to tell you something about the bacteria. And so some bacteria will cause this color change while others will not. And in this case, there's an indicator that's going to turn pink when bacteria ferment lactose. And so if bacteria can take this sugar, lactose, and use it in a metabolic pathway, fermentation, then what we're going to see is a color change that's part of the media itself. And so the end result then is the media, so the actual auger itself is going to turn kind of a hot pink color, or the bacterial colonies will appear a hot pink color. So let's take what we just discussed and see if we can kind of work some of these things out. So on this plate, you can see it's divided into 4 regions. And there are 4 different bacteria that have been streaked into each of the 4 regions. So starting with zone 1, what you want to do is ask yourself, is there bacteria growing there? And hopefully you can see, yes, indeed, there is this smear that's growing. And so remember what that means. If the bacteria grows, it's a gram-negative bacteria. And then if there is growth, you have to ask yourself, is the bacteria or the media turning hot pink? And indeed, while it's not super robust, you can see areas like here and here and here and here and here that it is indeed turning hot pink. So what we can conclude, there is growth. So this is a gram-negative bacteria. And it is pink as well, which means this gram-negative bacteria ferments lactose. So that's what we can conclude about bacteria in area number 1. In area number 2, same sort of logic that you go through. Is there bacteria growing? Indeed, there is. So the bacteria is growing. So it is a gram-negative bacteria. And then is the media turning hot pink? And hopefully you can see that this is a pretty clear hot pink of the media itself. And so we can again conclude there is growth. So this is a gram-negative bacteria. The media is turning pink, so this is a gram-negative lactose producer, or not producer, lactose fermenter. So it's lactose positive is another way that we phrase that. Looking at zone 4, it's a little harder to see, but you can see the zigzag here. So that's the bacteria growing. So we do have growth, so it's still a gram-negative, but in this case, the bacteria as well as the media itself is not turning pink. And so this is a gram-negative because it is growing in this 4th area, but it is not a lactose fermenter. So we'd say it's lactose negative. In zone 3, the bacteria is not growing at all. And so what we can say then is there's no growth. And so it's because the bacteria is gram-positive and being inhibited by those bile salts and crystal violet. And so this is how we can use this McConkey auger to evaluate bacteria. This is just, again, showing you these results. And so these are 2 different bacteria just to show you the difference in this hot pink color. So the bacteria on the right would be bacteria that do not ferment lactose. So as it's written here, non-lactose fermenting colonies. I don't know if I like that phrase, as well as bacteria or colonies that cannot ferment lactose. And then on the left, you can see that there is this hot pink color being generated where it is a lactose fermenting bacteria.